We wanted to see if the things we'd discovered, described, published, we'd come up with statistics and results. We wanted to verify them in actual practice, because we were, after all, already working on the biological level. Humans, animals, it's all biology. So, if we extrapolated everything we'd learned in the laboratory, using the same approach and the things we'd learned, we said to ourselves, we should be able to make it work with human beings.
Six months before his death in October 1998, Professor Mirko Beljanski spoke to us of his 50-year career as a researcher. It was his last major interview. To help us better understand his breakthroughs, we called on biologist Hervé Calvet, one of the researchers who worked with Beljanski in his final years.
I'm incredibly lucky to have been able to work with such an important researcher, one of the 20th century's great inventors alongside Marie Curie and other pioneers. Beljanski already had 40 years of research behind him and several major discoveries.
Avec des découvertes majeures.
My thesis centered principally on the effects of antibiotics. I thought this research would help me understand the fundamental substances of cells by studying bacteria and the mechanisms that govern cell life.
His research gave him a method for investigating and explaining various cell mechanisms by working inside the cells themselves, inside easily accessible cells, since it's easy to cultivate bacteria.
My thesis dealt with several different antibiotics. I showed that each of the antibiotics in question modified the behavior of the RNAs and DNAs.
L'ARN est ça veut dire acide ribonucléique.
RNA stands for ribonucleic acid. RNAs are very large molecules, very long chains within the cell. They're involved in the process of genome expression. In other words, in the expression of genes that are encoded by an even larger molecule called DNA, deoxyribonucleic acid.
L'ADN, l'acide désoxyribonucléique. Cet ADN
DNA stores information but can't express it directly. It's a little like, we could compare it to an enormous encyclopedia stored inside a very small library. A locked and closely guarded library with a very, very strict librarian who only lets information filter out from time to time, and only to the right people at the right time. When information is filtered out, obviously the entire library can't be moved. We have to make a copy of the page in question, and that copy is what we refer to as RNA. The copy simply contains the recipe for making, for synthesizing a protein. With the help of casseroles and utensils, we refer to as enzymes.
When I studied the chemical structure of shodomycin and compared it to the structure of the nucleotides in RNA, I found a striking similarity between shodomycin and one of the elements in RNA. We discovered absolutely amazing things that turned our way of thinking upside down, to such a point that at times we couldn't believe it ourselves.
Beljanski isolated bacterial mutants that had developed a resistance to the antibiotic shodomycin. He discovered that mutants able to resist the antibiotic could pass on that information to other bacteria. Those bacteria registered the information, developed a resistance to the antibiotic, and then passed along the characteristic in turn to their offspring.
We created mutants that could grow and multiply, just like the bacterial cells we cultivated in the presence of shodomycin. The structure of their DNA hadn't changed. Our tests verified that, but their RNAs had changed. That much was certain.
When a bacterium is exposed to shodomycin, it develops a resistance to the antibiotic. It undergoes a genetic transformation, a mutation that isn't triggered by DNA. The mutant bacterium then secretes an RNA inside the culture medium. This RNA penetrates another bacterium, inducing a genetic transformation which is then encoded in its DNA. This second bacterium in turn secretes another RNA.
He called these RNAs, which could transfer information, which could transform a bacterium that was sensitive to an antibiotic, in this case to shodomycin, and make it resistant. Beljanski simply called them transforming RNAs.
First of all, in the initial phase, we showed that the composition of the transforming RNA was in fact transcribed in the DNA of the bacterium that had received the RNA, but in the form of DNA.
Next, he identified the catalyzer, which made it possible to integrate the new information, which turned out to be an enzyme. RNA can't be directly transcribed in DNA, since there are two different types of molecules. The information that was stored in the RNA had to be recopied in a form that could be encoded in DNA. The information is transformed by an enzyme, reverse transcriptase.
We were able to show, later we proved it in test tubes, that this enzyme could copy the transforming RNA into DNA of the exact same length. Which means that the bacterium received the transforming RNA in the form of DNA.
This particular type of enzymatic activity, which doesn't normally occur in mammals, wouldn't be rediscovered until many years later. This time in viruses and in RNA retroviruses, most notably in the HIVs responsible for AIDS. He identified other types of RNA in human bacterial cells and in plant and animal cells. Even smaller RNAs that play a vital role in triggering the replication process. In other words, in triggering the copying of DNA. One of the very first and thus vital steps in cell division.
Une des premières étapes, donc indispensable pour la division de la cellule. Il y avait une certaine spécificité d'action.
These small RNA primers apparently played a very specific role. That gave Beljanski an idea. He decided to find out if he could stimulate replication and thus division in specific cells, but not in others.
Can we act on specifically selected targets, for instance on normal cells, in order to protect them? We wanted to regulate their genesis, especially the genesis of bone marrow. And most importantly, find a way to do just the opposite, but always acting selectively on specific targets, such as malignant cells, or on the DNA of malignant cells.
First of all, he had to find the material to do it with. So he took a very long RNA and cut it into fragments of uniform length. Now he had a large number of RNA primers to work with, making it possible to run experiments on animals as well as on cells.
We prepared small RNAs that can bind themselves to the DNA of bone marrow and induce the genesis of white blood cells, blood platelets, and later, red blood cells.
Et donc il a pu vérifier.
He verified that they did in fact act selectively. He also confirmed that there were even more blood cells afterwards, thanks to these molecules, while the number of cancer cells hadn't increased.
N'était pas surmultiplié.
This led to the birth of BLRs, which use Beljanski's famous RNA primers as their active principle.
BLRs are taken orally. They enter the bone marrow in just 20 seconds. We've been able to show that. They help bolster the immune system. They never weaken it. That's a proven fact. And they do it without any toxic effects.
Donc évidemment, l'application
BLRs clearly had a direct application, protecting blood cells from the aggressive effects of chemotherapy and radiation therapy, which are extrememely destructive for cells and for the entire immune system, at the very time the patient needs them most.
Avec ce concept déjà de RLB, eh bien, il avait déjà un premier moyen.
With the discovery of BLRs, we had a way to act selectively, a new tool in anti-cancer therapy.
La thérapie anticancéreuse.
In the beginning, no one was really interested in selective action. Researchers and scientists were still studying concepts that dated back 50, 60, even 100 years. Who knows? Substances referred to as anti-cancer drugs affected cancer cells and normal cells alike. Particularly the normal cells involved in the immune response, which were destroyed even faster than normal cells in other tissue.
Un rayon X ne peut pas
An X-ray can't target a specific site. In the same way, the substances used in chemotherapy don't act with any real specificity. We commonly refer to these substances as drugs. They enter all the body's cells without making any real distinction. Some are extremely toxic for malignant cells, but unfortunately they're extremely toxic for healthy ones as well.
How could we find substances that acted selectively, that affected only malignant cells and left normal cells unharmed? In the beginning, it was a fixation. We decided to take on the responsibility, if you like, of finding those substances. We said to ourselves, since substances with negative effects do exist and the lives and survivals of human beings are at stake here, then nature must also contain substances that have the opposite effect of the carcinogens that trigger cancers. Coming up with the idea was easy enough, but how were we going to find them?
In other words, the real problem was the tools. Beljanski came up with the fantastic idea of looking for new tools, instead of simply looking through the catalogue of molecules that had already been identified. But he had to find a way to discover new molecules. How did he do it? First of all, he had to find a specific structural difference between malignant cells and healthy cells.
We decided to start by verifying the behavior of the DNA in malignant cells compared to the DNA in normal cells, without having any idea of what we were looking for. We noticed right away that DNAs in malignant cells, whether they're from plants or humans or animals, we realized that these DNAs are always destabilized.
He used the word destabilized to describe a particular characteristic of this DNA. He showed that the DNA of malignant cells is broken at a certain point, instead of forming a stable and uniform double helix, without any breaks on either strand. Most importantly, he showed that the double helix is open, often along very long fragments.
Et ça sur parfois des très grandes régions. Et donc l'ADN qui se trouve
These fragments have a single-stranded DNA, and are thus highly accessible to all kinds of molecules, including enzymes, which turned out to be extremely useful for DNA replication and for triggering the division of cells.
There is a clear-cut difference between the DNA of malignant cells and the DNA of normal cells. At that point, we had the basic material to work with. Next, we had to find a substance that acted selectively on malignant cells. That prompted us to develop a highly sensitive and rapid biochemical test. We get results in just 2 to 3 hours, telling us if a given substance acts selectively on the DNA of malignant cells and not on the DNA of normal cells.
Et en fonction de son test, de l'oncotest,
In view of the test results, he set his criterion that nothing happened to the DNA of normal cells, but in malignant cells, in contrast, DNA replication was halted, which meant that the cell couldn't divide and would die. Mirko Beljanski was ahead of his time. Today, the pharmaceutical industry uses the same process. It's known as screening. In other words, they develop a test, take every molecule they can find, then run them systematically through the test. Beljanski, for his part, took a special interest in plants, which offer a tremendous reservoir of molecules.
If non-toxic substances can be found in plants, then it's worth our time to try and find them, track them down, and isolate them, because these substances have already learned how to survive in a living cell. Even if it's a plant cell, it's still a living cell. Finding a normal biological material that meets your criteria is a tremendous advantage, because chemistry may be very powerful. Nonetheless, it's still extremely difficult to create molecules that strong through chemical means. We found an extract from an African plant that had 52 different alkaloids, which are particular substances found in plants. We very quickly identified one that met our criteria, a molecule that must have said to itself, "You found me at last, and now I've found you."
He identified two very interesting molecules, one in a plant from Africa, from the equatorial forest in Africa, and another in a plant from the Amazon forest. The first was alstonine, an alkaloid he isolated from the 52 different alkaloids of the Rauwolfia vomitoria plant. He isolated the other molecule, known as flavopererine, in the pereira tree that grows in the Amazonian forest. Both molecules met his criteria perfectly.
We discovered two different substances at almost exactly the same time. We established that exposure to these substances, each of them separately, destroyed malignant cells, but the substances had no effect on normal cells.
He got the same result every time. In other words, the malignant cells were killed, but not the control cells. The non-cancerous cell from the same tissue, for instance normal lung cells compared to malignant lung cells, were unaffected by the presence of the alkaloid.
Next, we had to find out why they acted selectively.
Nous savons que la membrane
We know that the membrane of malignant cells has a negative chemical charge,
négativement,
and that the membrane of normal
membrane de la
cells
cellule normale
has a
charge
positive charge.
positivement.
We
La
were very
chance
lucky
que nous
in
avons
that
eue,
these
c'est que les
lock
molécules
molecules,
verrous que nous
we
avons
call them
appelées
lock
verrous,
molecules, or
anticancéreuses
specific
spécifiques,
anti-cancer
ont
molecules,
deux charges,
have two charges,
une charge positive, une charge négative.
negative charge and the other positive.
Donc, voyez-vous, avec
So,
une charge
with
positive et négative,
a negative and a
ces
positive
substances
charge,
pouvaient sélectionner
these substances can identify malignant
les
cells,
cellules cancéreuses, entrer dans
enter
la
them,
cellule cancéreuse,
and then move through the
traverser le
cytoplasm
cytoplasme
and
et
go
se
straight to the
diriger
nucleus.
directement vers le noyau.
These lock molecules don't penetrate healthy cells, as this image clearly shows. Flavopererin, which appears as a yellow halo under normal light, surrounds the cell but remains outside. The malignant cells, meanwhile, are penetrated by the same concentration of pereira. The product goes directly to the nucleus, concentrated in the nucleolus. The blue fluorescence visible under ultraviolet light shows that the molecules have penetrated the cell.
They manage to slip through, entering the nucleus of the cell where the DNA is stored. There, in the presence of the extremely active destabilized DNA, they strike their target.
Elles se bloquent.
They lock
l'ADN,
onto the
l'ADN
DNA,
ne peut pas être
blocking
bloqué.
it.
Donc,
The DNA can't replicate, which means that the cell will die, because cells can't live without DNA replication.
de l'ADN. Ces deux molécules,
The two molecules we developed, if you would, lock onto simple chains of DNA and try to bring them together.
il essaie de les rapprocher,
They form a lock of
faire une
sorts.
sorte de verrou.
The chains can no longer
Et les
breathe,
chaînes ne peuvent
as I've
plus,
explained in my
comme je
conferences.
disais quelquefois dans mes conférences, ne
They
peuvent plus
can't
respirer. Ne peuvent pas
open and
s'ouvrir et se
close.
fermer. Ces molécules se fixent.
These molecules lock onto specific DNA sites. We've been able to show that these sites are all of the same type. They're all composed of the same kind of nucleotides, the same basic structural units of the DNA in malignant cells.
Ceci est très important.
This is extremely important. Science has had a hard time explaining why there are 30, 40, 50, even 100 different types of cancer.
40 ou 50 ou 100 cancers différents. C'est vrai que les cancers sont différents.
It's true that cancers are different,
Nos gènes,
but
nos
human
chromosomes,
genes and chromosomes are always the same in every type of
Et
tissue.
si une cellule
So if cells from a prostate,
de la prostate,
or a brain,
du cerveau,
or a
du
liver,
foie,
I'm
des
talking
cellules
about
cancéreuses, j'ai dit,
malignant cells,
va répondre
all
à
respond to
ces
these lock molecules.
verrous. Ça veut dire
What does
quoi?
that tell
Ça veut
us?
dire que il y
It
a
tells that there are specific common sites shared by
comme
all
qui
types
se
of cancer.
trouve dans tous ces cancers, dans les zones.
The differences between the various types of cancer are due to the fact that each organ or tissue has a different structure, a different physiological mechanism.
Le
But
fonctionnement
the
physiologique,
genome is
mais le
always
génome est
the
le
same.
même. L'ouverture des chaînes d'ADN est
The
quelque
opening
chose
in the
d'extrêmement-
DNA chains
d'extrêmement-
is extremely,
quelque chose d'extrêmement important
extremely
parce que
important,
ça-
as it makes it
ça
possible
offre en quelque
for lock molecules
aux molécules
to
qui
bind
sont dans la cellule
to the DNA.
de prendre place sur cet ADN. Et
The separation
on peut
of the
aussi
DNA
penser
chains
que la séparation des chaînes
is
d'ADN est
also
absolument
of vital
utile
importance for these
pour les
substances,
substances ou les
which
produits
act selectively on the
qui
DNA
agissent
of
sélectivement
malignant
contre
cells.
l'ADN des cellules cancéreuses.
Il s'est dit,
He
mais
asked himself
peut-être que
if these molecules, flavopererin and alstonine, have a particular affinity for destabilized DNA,
particulière pour l'ADN déstabilisé.
then would they become more active if the DNA was that much more destabilized?
Active, tout simplement.
Les ADN déstabilisés
When exposed to chemotherapy or radiation therapy, destabilized DNAs become even more destabilized.
déstabilisent encore plus.
Consequently,
Et dans ce
since the
cas-là,
product acts
comme le
only on
produit
malignant
des cellules
cells,
cancéreuses,
it
il va pénétrer
penetrates
beaucoup
those
plus
cells
rapidement dans ces
even
cellules.
quicker. It works in synergy with chemotherapy or radiation therapy, locking onto and helping destroy malignant cells much more rapidly and more efficiently than with chemotherapy alone, or radiation alone, or the product alone. Patients who undergo radiation therapy suffer tremendously.
He went straight to the heart of the problem. X-rays are harmful. Perhaps nature contains something that can help us combat their negative effects.
We identified several different substances that could protect animals against radiation, including a plant extract.
He learned that life had gone on after the Hiroshima disaster, that one plant, in particular, had survived and continued to grow in Hiroshima, the Ginkgo plant. He thought to himself, well, if this plant could be subjected to incredibly strong radiation and continue to grow afterwards, then it must have something special, a way of defending itself, a way of fighting against the harmful effects of radiation.
Ginkgo is a biological regulator. It can also preserve certain metals, keeping them at the disposal of mucous membranes if there is a sufficient quantity of the product in the intestine, for instance.
He took a group of rats and exposed the poor things to extremely high doses of radiation, doses which should have killed them, but they remained in perfect health, thanks to the molecule he'd isolated in the Ginkgo biloba plant. He even checked their DNA chains and confirmed that there were no breaks in their chromosomes. In other words, their DNA was intact.
If this plant was able to resist the atomic bomb, don't forget that, then that means it had an entire arsenal of weapons, which means, if it's used the right way, it could work wonders.
Madeleine Piloget has breast cancer. 5 years after her operation, her cancer returned. She's being simultaneously treated with chemotherapy and several of Beljanski's products. I take a very strong dose. Every day I take 4 PAO-FMs, 4 PAO-5s, 4 Ginkgo bilobas, and 4 Rauwolfia extracts, plus 1 real build every other day. This chemotherapy is extremely toxic for your blood count. My red blood cells have stayed at a normal level, but my white blood cells fluctuate wildly. So much that they couldn't give me the second round of chemotherapy because I didn't have enough neutrophils. But since then, everything's gone just fine. I should tell you that the week before the second round of chemo, I lost my pill blocks in Blois. I stupidly dropped the 2 little doses of real build I was supposed to take into the gutter. And the effect was immediate. My blood count nosedived, and I couldn't go ahead with my chemotherapy. My chemotherapy is a mix of FEC50, fomorubicin and doxin, and 5-FU.
5-FU, c'est quand même...
It really is a highly toxic dose.
Oui, oui. Et je suis étonnée.
And I'm surprised.
Étonnée.
To be honest, I'm truly surprised that doctors go ahead and prescribe things that destructive without a second thought, because they know perfectly well they're injecting toxic products into our bodies. And I wonder what they would do if it were them. I've gone through chemotherapy without any side effects. I've never been nauseous. They went ahead and prescribed Zofrene anyway, but I just cut the pills in 2. I lead a perfectly normal life for someone my age. I drive myself to the clinic, it's around 55 miles, and then I drive back again the very same day. Then I go take care of my 5 grandchildren, because their mother gets home from work late.
The same day you take your chemotherapy?
Oh yes, I drive right back home. And I see a lung specialist on a regular basis, and he's been amazed. He even asked me if he could prescribe Beljanski's products.
Le chirurgien.
Then the surgeon at Dublanc Hospital can't get over me, because he operated on his own sister for breast cancer 5 or 6 years ago, and she's been dead for quite a while.
Ah, moi, je m'en
I'm
sors,
beating
j'en
it.
suis persuadée.
I'm convinced. 100%.
J'ai une vie
I have
intéressante.
an interesting life,
Je tiens pas à
and
la
I don't
perdre.
want to lose it.
His approach to diseases caused by viruses echoed the work he'd already done on bacteria cells, animal cells, and malignant cells. He kept on exploring the same line of research and a very promising specific intrinsic target, the DNA of diseased cells and their destabilized structure. By studying the genome structure of certain viruses, he was able to show that their nucleic acid, either DNA or RNA, since some viruses encode their information in the form of RNA instead of DNA, he showed that the nucleic acid of these viruses, which functions as their genetic material, had a structure with strong similarities to the destabilized structure of malignant DNAs.
On s'est aperçu assez rapidement qu'il y en avait
We
une
noticed very early on that one of these molecules greatly hindered the replication of these viruses
application de ces virus
in
chez
animals.
les animaux. Nous avons
We decided to verify at once
sur
on 3 different levels. First of all, with plants like tobacco. We started with the tobacco mosaic virus. We coated the entire surface of a leaf with the virus, then divided the leaf in 2. We put the product on one side and nothing on the other. On the side that hadn't been treated with the antivirus product, the virus continued to replicate. On the other side, the virus disappeared, or there were just a few wheels left because we hadn't used a high enough concentration of the product. The influenza virus is a different type of RNA virus than plant viruses. We can inject the virus into mice through the nose. The virus reaches the lungs, then invades the lobes of the lungs. In the mice that had been given the test product, the same one we'd used on tobacco, we confirmed that there were no traces of virus in the lobe or lobes, if you prefer, which means the virus wasn't replicating. When AIDS came along, it was perfectly logical for us to verify what the product could do in animals and eventually in humans.
Gerard Weidlich was a lifeguard. In 1985, he performed mouth-to-mouth resuscitation on a drowning victim who was vomiting blood. Weidlich was infected with the HIV virus, which rapidly developed into AIDS.
I noticed the first symptoms in the winter of 85-86. At first, I was totally worn out. Then I started suffering constant diarrhea. I developed an explosive case of recurrent herpes and a yeast-like fungus that spread over my entire body. I went to see my doctor and he sent me to Professor Mirko Beljanski, since he was totally powerless to fight my disease.
Puisqu'il se trouvait tout à fait impuissant face à cette maladie.
This was back in 1985, when there wasn't any treatment for this deadly disease. I went to the pharmacology department. Beljanski made the very first capsules of his molecules for me, under the code name PB100. So I started taking his products on April 15th, 1986. And my herpes, which, as I said, had completely exploded, was brought under control. The opportunistic diseases I mentioned progressively disappeared, one by
ont
one,
disparu progressivement, les unes
and I
après les
haven't
autres,
fallen
et
ill
je ne
since.
suis jamais plus tombé malade. En septembre
In
81,
September of 91,
et j'ai fait faire
they
un
ran a
examen
PCR
à
test on
l'Institut
me
Pasteur,
at the
de
Pasteur Institute in Paris.
Pasteur.
It's a very important test that confirms the presence of
Et c'est un
provirus
examen extrêmement
DNA.
important puisqu'il recherche la présence
And
du
luckily,
gène
I've got the exam results in my possession today, because if I didn't, I'm sure people would say I had never had AIDS. Why? Because after developing full-blown AIDS symptoms, the disease has been in recession for 13 years. The product isn't toxic. Because if it was, given the incredible number of capsules I've ingested over the years, I would have died a long time ago. I weighed 160 pounds when I was contaminated. I've gained weight since. Even with a full-blown case of AIDS, today I weigh just under 200 pounds. I don't use the word cured. I'm more prudent than that. But I do believe that everyone who has been struck down by this deadly disease should be able to go on living a comfortable life, the way I have.
Exceptionnel comme je peux en bénéficier moi-même.
Francine Bouquet was infected with the AIDS virus in 1984 during a blood transfusion. 4 years later, she was in the final stage of the disease.
4C2.
I was in 4C2,
Alors ça veut dire
which means I didn't have much longer to
longtemps à
live.
vivre. J'étais
I was a
fichue.
goner.
Et là, j'ai pris
And at that
conscience
point,
qu'il fallait que je
I realized I
prenne
had to take my health in hand. Everyone is always telling you to take responsibility for your own health. That's exactly what I did.
Quand j'ai pris le PB100,
I first started taking PB 188,
au bout d'un
and
mois,
after just 1 month, I
j'ai
felt
une
better.
amélioration. La fatigue que
I wasn't
j'avais
tired all the time like before.
Les maladies opportunistes,
The opportunistic diseases gradually disappeared.
disparu petit à petit. J'avais moins de fatigue.
I felt less
Ça,
tired.
ça, c'est
It was really, really amazing.
J'avoue que je sentais un dynamisme
I had newfound energy,
que je ne ressentais
something I
plus
hadn't felt for over 4 years.
de refaire du sport.
I started doing sports again, swimming and playing tennis.
C'était pas
There's just no comparison.
Je suis même obligée
I've
de
even
faire,
had to go on
des régimes.
diets.
J'ai pris 9 kilos.
I've put on almost 20 pounds
Aucun effet.
with
Y a
no
pas de
side effects.
toxicité.
They're not toxic. I'm fine.
Je n'ai rien.
They're
Je
easy to
digère
digest.
bien,
I can
je peux
take
les
them
prendre,
anytime.
J'ai pas d'heure précise.
I don't set an exact time.
Je les
I take
prends
them around noon.
même le midi.
And if I forget them, it's really no big deal. I take them later that night. I don't have to take them at a set hour. I'm not a vegetable.
J'ai 14 ans.
The disease has been in recession for 14 years. That includes 10 years taking Beljanski's products.
14 ans de recul.
14 years.
Je viens d'avoir
And I just gave birth to a little girl
Eugénie.
named Eugenie.
Et c'est vrai que je prends conscience que,
And I realize now that if I hadn't discovered these products,
comme j'étais
since I
qu'à
was
la
already in the
C2,
4C2
bon,
phase,
je l'aurais pas
I
vu.
would never have had her.
On prend conscience quand
You
même que la vie
realize
est
just
belle
how beautiful life really
quand
is
on a failli la
when you've
perdre.
almost lost it.
Je suis
I'm
biologist.
a biologist.
J'ai fait des études de
I've
chimie
studied both
biologie.
chemistry and biology.
Ma spécialité
My
est
speciality is cellular biology,
et toutes mes
and
recherches
I've conducted all my research with my friend and colleague Dr. Philippe Anker.
Philippe Anker. Nous avons
We started
travaillé
working together in 1962 and have worked together without fail ever since.
fait ensemble. Nous avons émis l'hypothèse
We hypothesized that DNA exited cells, circulated, then entered other cells, and could even have a direct effect on them. It was a gamble.
Nous allons, c'est en
We
62
put forward that
cette
hypothesis in
hypothèse, nous allons
62,
découvrir seulement
but we didn't confirm that
73
DNA did in
ADN
fact circulate until
Et
73.
il faudra attendre 96
Our work wasn't recognized until
soit
1996,
reconnu.
Not until our research on circulating DNA prompted us to take a closer look at cancer. We linked this phenomenon to a problem in cancer research, the detection of cancer. DNA circulates, which means that DNA also leaves malignant cells. Consequently, we no longer need to perform a biopsy. All we have to do is take a sample of 5 or 10 milliliters of blood, then look for malignant DNA in the blood plasma. It's a non-invasive way of detecting cancer with one considerable advantage. After an operation, for example, you can run tests every 2 or 3 months to check for metastasis.
Voir s'il n'y a pas des métastases qui se forment. Pour voir si
we used Beljanski's discovery to look for malignant DNA in blood plasma.
Dans le plasma du sang, nous avons utilisé la découverte de
Would we find DNA with the structure described by Beljanski in blood plasma?
qui avait les structures décrites par
That is, with unwinding DNA,
des ADN
destabilized DNA.
et nous avons
That's exactly what we found.
Il y a deux découvertes
We worked on 2 of Beljanski's discoveries.
sur lesquelles nous avons travaillé nous-mêmes. Nous avons dit que
We
l'ADN
confirmed
sort
that DNAs do leave cells,
mais il y a aussi un ARN.
but an RNA leaves as well.
Et nous avons découvert que
And we
justement,
confirmed something else.
les ARN qui sortent sont
The
des
RNAs that
ARN,
leave
les
cells are the
ARN
small RNA primers discovered by Beljanski.
Nous avons d'ailleurs publié
We published our findings in a major scientific review, Cancer Research,
soulignant que
underscoring the fact that Beljanski's RNA primers also left the cell along with our DNAs.
nos ADN. Actuellement,
At present,
de vrais médicaments
there's only one real substance that combats the harmful effects of chemotherapy and radiation therapy, the BLR.
amide qui
A friend of mine was diagnosed with a terrible case of leukemia not long ago.
grâce au
Thanks to BLRs, he was able to ask his doctors to continue his chemotherapy even longer than usual, which
normalement
increased his chances of survival.
augmenter beaucoup ses chances de survie. C'est le seul
It's the only
candidat.
product that makes that possible.
Et qui a
And there
aucun
aren't
effet
any side
secondaire.
effects.
C'est
It's
ça qui est
fantastic.
fantastique. Les traitements de Beljanski
You can take Beljanski's anti-cancer products for years.
anticancéreux, vous pouvez prendre ça pendant des années. Moi, j'ai des amis.
I have friends with cancer who have taken Beljanski's product every day for 5 years
tous les jours,
without any side effects,
et sans avoir aucun effet secondaire. Parce que
because
c'est un
it's a
produit
specific product.
C'est ça
That's
la
the
beauté
beauty
du
of the
produit.
product.
En disant, qu'est-ce que c'est
What is
un
it
produit
exactly?
de Beljanski contre le cancer.
It's a non-toxic form of chemotherapy you can keep taking forever if you want to.
Éternellement, si ça vous amuse. J'avais été
I
très,
was
très
extremely
impressionné
impressed by the
par
work
les
he had
travaux
published.
qu'il avait publiés, où il montrait
He
qu'un
gave
de
certain
certains
types of
alcaloïdes,
alkaloids to mice
sur des souris
that had been infected with cancer,
son produit permettait
and close to 40% of the mice that used his product survived.
la chimiothérapie
With
seule
chemotherapy alone,
permettait à
50% of the mice survived.
aux souris de survivre, mais la
But the mice that were treated with both chemotherapy and his product had a survival rate between 80 and 90%. When you read about his work in the writings on cancer, the results are very, very impressive.
Alors à Genève,
An oncologist in Geneva, Professor Maurice, ran tests on malignant human cells and on cancer cells that resisted
Des
chemotherapy.
cellules cancéreuses qui étaient résistantes à la chimiothérapie.
He discovered that these cells responded to one of the alkaloids used by Beljanski, and that the growth of cells was blocked.
Sensible à cet alcaloïde, donc ces souches, le développement de ces souches était bloqué. C'est un résultat
It was an important breakthrough.
important. Alors là,
In view of these results, Beljanski very courageously went ahead
ayant de tels résultats,
and advised cancer patients to take his alkaloids.
à des cancéreux de prendre ces alcaloïdes. S'il avait
If he
tué
had
des
killed
malades, on aurait
anybody,
dit qu'il était fou.
people would have said he was crazy. But the patients did very well, so it's fair to say that he's a man of true genius and courage.
Beljanski est un
I
chercheur
think that Beljanski is an amazing researcher.
assez étonnant. Bon, premièrement, je dirais que c'est un
I'd call him a finder above all else.
rien dire.
Being a researcher doesn't really mean much. You can spend a lifetime searching. You have to find something, and Mirko Beljanski found a great many things. It's very surprising. He made so many fundamental discoveries and published his findings in hundreds of reviews, very important reviews. But since the Pasteur Institute had barred his route in France, his work never made a big impact.
Ses travaux n'ont pas marqué, que jamais.
No one has ever criticized his findings in a scientific review, and knowing that France wanted him out of the way, I'm sure they must have verified whether the findings he'd published were true or not, and when they realized it was true, they decided there was no point continuing.
Vous savez,
You know, when you make a discovery, that discovery has to be discovered in turn.
soit découverte.
Ce sont des choses qui, dans l'avenir, doivent se développer.
Things should start moving forward in the near future.
Maintenant, par des-
If they're delayed now because of the adverse conditions we're facing, no, we're going to see real headway.
commence déjà à l'étranger.
People are starting to show interest abroad to get things moving. It may take a little time, but the wheels are already in motion.
Mais maintenant, c'est parti.
The torch has been passed on to the United States, where a team of young researchers is now pursuing Mirko Beljanski's pioneering research program.